Yes, today we are going to see the Kreb cycle as the third step in the aerobic respiration. Step 1, it is glycolysis. We know during the process of glycolysis, what happened? It is nothing but the glucose, the respiratory substrate glucose. It is broken down into two molecules of pyruvic acid during the process of glycolysis. Now, this pyruvic acid, which is contained in 3 carbon, it undergo link reaction. And during the link reaction, the first thing that happens to pyruvic acid, 3 carbon, it undergo decarboxylation. And during decarboxylation, one molecule of CO2 will be removed. During oxidation, hydrogens will be removed and they will be taken up by NAD to form NADH2. As a result of this, we know a 2-carbon compound is produced, and that is called as acetyl moiety. That is called as, we can say, acetyl molecule. Now that acetyl molecule will combine with coenzyme A on the outer surface of mitochondria, and then there will be formation of acetyl-CoA, which is containing 2 carbon, and this 2-carbon compound can very easily pass through the mitochondrial matrix. And once it enter into the mitochondrial matrix, again that acetyl-CoA will undergo hydrolysis. Again, there will be formation of CoA, which will be available for bringing the other carbon acetyl moiety into the mitochondrial matrix. Once that is done, now that acetyl moiety, it will be present within the mitochondrial matrix. And this acetyl moiety containing 2 carbon, it will combine with a 4-carbon oxaloacetic acid in the mitochondrial matrix. Again, I am repeating the entire Kreb cycle. It is occurring within the mitochondrial matrix. Okay, so in the mitochondrial matrix, a 4-carbon oxaloacetic acid, it is the acetyl moiety acceptor. 2-carbon acetyl moiety combines with 4-carbon oxaloacetic acid. 4 plus 2, it will be the 6-carbon compound which is produced. That is nothing but the citric acid. And this citric acid, it is the first stable compound of the Kreb cycle. That's why Kreb cycle is also called as citric acid cycle. Right? Now this citric acid, 6-carbon citric acid, it undergo dehydration, removal of water molecule. And by removal of water molecule, this citric acid, it is converted to cis-aconitic acid. Again, same carbon, carbon मध्ये काय change नाही आहे. So it will be the 6-carbon cis-aconitic acid will be produced. Now this cis-aconitic acid, again it undergo hydration, addition of water molecule, and it will be converted to isocitrate. So that is the step 1, where 2-carbon acetyl moiety, it comes into 2-carbon acetyl moiety enter into the mitochondrial matrix. In the mitochondrial matrix, 4-carbon oxaloacetic acid is available. Now this 4-carbon oxaloacetic acid, 2-carbon acetyl-CoA, they will combine together to form 6-carbon citric acid, and that's why the cycle is called as citric acid cycle. Now this 6-carbon citric acid, it undergo dehydration, and this dehydration will result into formation of cis-aconitic acid. Cis-aconitic acid undergo hydration again to form isocitrate. That is the step 1. Now this isocitrate, now it undergo oxidation. Oxidation bole to removal of hydrogen. That hydrogen will be taken up by NAD, and there will be formation of NADH2. And by oxidation of isocitrate or isocitric acid, there will be formation of another organic acid. Now that another organic acid, we call it as an oxalosuccinic acid. Now this oxalosuccinic acid, it is containing how many carbon? 6 of them. Now this 6-carbon oxalosuccinic acid undergo decarboxylation. Now what is decarboxylation? Removal of carbon dioxide. So carbon dioxide will be removed from the 6-carbon latecomers. Yeah. From the 6-carbon oxalosuccinic acid, 1 CO2 will be removed. So 6-carbon oxalosuccinate will be converted to 5-carbon organic acid. And that 5-carbon organic acid, it is called as alpha ketoglutaric acid. It is called as alpha glutaric acid. Now this 5 carbon alpha glutaric acid, it undergo oxidative decarboxylation. Oxidative, hydrogen will be removed. Decarboxylation, CO2 will be removed. Now during the oxidative decarboxylation of alpha glutaric acid, hydrogens will be removed and they will be taken up by NAD to form NADH2. And during decarboxylation, one molecule of CO2 will be given out. Now that molecule of CO2 will be coming out of the alpha glutaric acid, which is containing 5 carbon. Automatically from the 5, one CO2 goes and 4 carbon unstable moiety molecule will be produced. Now that 4 carbon moiety, it is called as succinyl moiety, succinyl molecule. Now this succinyl moiety, once again it will combine with coenzyme A and there will be formation of succinyl CoA. Now this succinyl CoA, it undergo hydrolysis. It will be undergoing hydrolysis, addition of water will be there, and as a result of that, succinyl CoA will be converted to succinic acid. Now during this process, one molecule of GDP, guanosine diphosphate, will be converted to guanosine triphosphate. But this guanosine triphosphate, it is equivalent to ATP. GTP and ATP are having the same value, energy value. But during that hydrolysis of succinyl CoA, there is a formation of 4 carbon succinic acid. Now this succinic acid, 4 carbon, it again undergo oxidation, removal of hydrogen. The removed hydrogen will be now accepted by another coenzyme that is called as flavin adenine dinucleotide. Flavin adenine dinucleotide receives the hydrogen from succinate and it is converted to FADH2. During this process, by oxidation of succinic acid, there is a formation of another organic acid that is called as fumaric acid. It is called as fumaric acid, right? Now this fumaric acid, it undergo hydration to form malic acid, another organic acid that is called as malic acid. Now this 4 carbon malic acid, it undergo oxidation, removal of hydrogen. Removed hydrogen will be taken up by nicotine amide dinucleotide NAD, which is converted to NADH2, and that malic acid is converted to oxaloacetic acid, which is available for repeating the process. So again, oxaloacetic acid will be ready. Again, it will take up the acidyl moiety. Again, it will go through the entire metabolic pathway. That is how the Kreb cycle, citric acid cycle, it goes. Now here you can see the citric acid, cis-aconitic acid, alpha glutaric acid, even the fumaric acid, malic acid, all of them are containing 3 COOH group, that is called as carboxylic acid group, and that's why it is also called as tricarboxylic acid cycle. Tricarboxylic acid metabolism. That's why it is called as TCA cycle. And the great scientist Sir Hans Kreb was the first one to discover this cycle in the pigeon muscle. That's why it is also called as Krebs cycle. And for that purpose, Sir Hans Kreb received the Nobel Prize, that already we know. So this is the way how the Krebs cycle is working, right? Yes, it is the net gain of the Krebs cycle, or it is the balance sheet for Krebs cycle. Now during the Krebs cycle, we know how many of the NADH2 are produced. Let's see there. Here. Now you can see during the oxidative decarboxylation, आता इथे 2 molecule pyruvic acid चे. So there will be formation of 2 NADH2 here. Then you can have 2 NADH2 here. Again 2 NADH2 here. Again 2 NADH2 here. So how many of them? 1, 2, and 3. And during oxidative decarboxylation 2. So totally 8 NADH2 are produced and 2 FADH2 are produced, and again 2 ATP will be produced. Now this is what we can say the net gain. Now that net weight gain, we can say during the glycolysis, during the Krebs cycle, 6 molecules of NADH2 directly. Now that 6 molecule, each will produce 3 ATP. So total will be 18 ATPs that are produced during Krebs cycle. 2 molecules of FADH2. From 1 molecule of FADH2, 2 ATP will be produced. So 2 into 2, totally 4 ATPs. And 2 GTP means 2 ATP. They are equivalent. So there will be formation of 2 ATP molecules. 18 plus 4 plus 2. It is equivalent to 24 ATPs that are produced only during Krebs cycle. And for this generation, once again, 6 NADH2 produces 18 ATP. 2 FADH2 produces 4 ATP and 2 GTP produces 2 ATP. Why? Because 1 molecule of NADH2 gives 3 ATP, 1 molecule of FADH2 produces 2 ATP, and 1 molecule of GTP equivalent to 2 ATP molecule. That is a way we can say net gain as far as the Krebs cycle is concerned. Again, I will go and show you where the ATPs are generated. Here you can see oxidative decarboxylation that produces 2 ATP. जो नंतर आपण हिशोब करणार आहे. आणि actual Krebs cycle मध्ये here 2 here 2 here and 2 here, totally 6 NADH2 are produced and 2 FADH2 are here and 2 ATPs are from 2 GTP. So that will be the 3, 6, 9. Okay. So 2, 6 into 3. 6 3 is 18. 18 then 4 here and 2 here. So there will be formation of net 24 ATP molecules during the Krebs cycle only, and you can add 6 ATP from the oxidative decarboxylation. It will make 30. Okay, and 8 molecules during the glycolysis, they will make 38 ATP. So that will be the total net gain during the process of aerobic respiration that involve glycolysis, that involve oxidative decarboxylation and Krebs cycle. However, if we go for the significance, what are the important features of this Krebs cycle? Number 1, it is a common pathway for breakdown of carbohydrate, fat, amino acid. Number of times I have told you, if glucose is available, it will be the respiratory substrate. If glucose is not available, fatty acid will be used. Even the fatty acids are absent, amino acid can be used for generation of ATP. So preferred respiratory substrate is glucose. If glucose is not available, fatty acid. If fatty acid is not available, then even the amino acid can be used. That is the way. Then it is a major pathway for generation of ATP molecule, which make energy currency of the cell. Already we know that this process of ATP generation, it can give us 30. Yes, this Krebs cycle, it is a major pathway for generation of ATP molecule, which make energy currency of the cell. Totally, from 1 glucose molecule, we will obtain 38 ATPs, and that is quite big amount. So it is a major pathway. Then it forms so many intermediate compounds, the different variety of organic acid which are produced, they will be used for synthesis of other biomolecules like amino acid, like nucleotide, like chlorophyll, the cytochromes, and even the fat. So all the organic acid which are produced as an intermediate compound, they can be used in the other biosynthetic pathways. Not only that, during the Krebs cycle, amino acid can be synthesized directly from alpha ketoglutarate acid containing 5 carbon, pyruvic acid containing 3 carbon, and oxaloacetic acid containing 4 carbon. So 3 carbon pyruvic acid, 4 carbon oxaloacetic acid, and 5 carbon alpha ketoglutarate acid can be directly used for synthesis of amino acids. Now these amino acids will be further used in the synthesis of proteins or even the nucleic acid, then the nitrogen bases that we know, purines and pyrimidine. So many intermediate compound can be produced from this Krebs cycle product. Not only that, The acetyl-CoA, which is a transporter molecule, it is related to the synthesis and breakdown of fatty acid, steroids, carotenoids, then terpenes, and even the aromatic compound, they can be produced from the acetyl-CoA. Then the next compound that was produced as an intermediate, it was succinyl-CoA, which gives rise to succinic acid later on by hydrolysis. That succinyl-CoA, it carries synthesis of pyrrole compound and pyrrole aromatic ring compound, which are required for production of chlorophylls and cytochrome. So chlorophyll and cytochrome, these are the aromatic compound which require presence of pyrrole, 5-carbon organic ring, aromatic ring. It can be produced from succinyl-CoA. Then it forms a number of reduced coenzymes like NADH2 and FADH2. NADH2 and FADH2, these are the hydrogen acceptor coenzyme which get reduced. And later on, in the next step, that is electron transport system, they can generate the ATP molecule. Already we know NADPH2 3 ATP, FADH2 2 ATP, that will be produced during the electron transport system. Then this Kreb cycle, it also involves oxidative breakdown of fats and protein. A little bit of repetition point that for Kreb cycle, not only glucose, but even fats and protein can be used. And the last thing, the intermediate compound which are formed during Kreb cycle are used for synthesis of nucleotide as well as amino acid. So these are the significances of Kreb cycle, and this Kreb cycle, it is a major pathway as far as the generation of energy, generation of ATP molecules can be observed in the biological world. Okay, so that is story of Kreb cycle.